The overall goal of this procedure is to obtain the skill and knowledge to perform the purification of DNA from a plant that was cut from an agarose gel by melting the agarose with the subsequent use of a purification column. Nowadays, there's the need to recover and purify DNA fragments separated by agarose and polyacrylamide gel matrix by means of gel electrophoresis, so that multiple nucleic acids purification methodologies can be well managed. Thus, a specific DNA fragment regarding its respective length purification turns out to be useful when having multiple DNA fragments in a sample and only one of them is worth for us. For example, when more than one band is visualized in an agarose gel electrophoresis after having executed a PCR reaction, or even more when carrying out a plasmid enzymatic digestion to subclone a fragment in a second vector. Besides, it is important to purify DNA so that processes that depend on nucleic acids quality and purity, such as PCR reaction, enzymatic degradation, and cloning, can be well executed without interfering with enzymatic activity and process development. In other words, an adequate nucleic acid extraction, selecting the adequate lysis, separation, and precipitation methodology, as well as the correct management of reactants such as DNTPs, primer features, DNA, RNA, template amounts will about protein and cell contamination, joined with enzymatic activity, inhibition, and unspecificity.
There are multiple purification methodologies regarding nucleic acids. Electrolysis through DNA recovery in a cell trap. The second one, electrolysis through DNA recovery in a dialysis bag. The third one, electrolysis through DNA recovery in a membrane. The fourth one, agarose degradation through agarose enzyme. The fifth one, hot phenol. The sixth one, magnetic bead purification. The seventh one, DNA fragment purification with GTG agarose. And the eighth one, silica columns.
There are many DNA purification kits by columns such as Ultra Clean 50 DNA Purification Kit, Simon Clean Gel DNA Recovery Kit, Kit Pure Link, that are described in the table.
Regarding DNA purity, for DNA fragments equal to 15 kilobase per or more, it's preferable to use the electrolysis through DNA recovery in a membrane. This method has the advantage of obtaining quality in DNA, but the range of reaching quality starts at 5 kilobase per. On the other hand, DNA purification for 1 kilobase DNA fragment, it's highly recommendable to use DNA purification by means of silica columns.
The purified DNA sample, 30 microliters, was loaded in the gel and run until the pans, 110 volts for 60 minutes approximately, were properly separated using 4 microliters of loading buffer. The gel was visualized on a transilluminator and the corresponding band was cut to the DNA of interest, trying to have the least amount of agarose possible. The gel fragment was cut into a sterile 1.5 microliters microtube. The rest of the gel was disposed at the right container. Purification kit with Pure Link. The cut gel fragment was weighted and 400 milligrams per 1.2 milliliters were added with buffer L3. The mixture was placed in the bath at 50 Celsius degrees for 15 minutes at intervals of 3 minutes, mixed by inversion 5 times to ensure the dissolution of the gel. The mixture was placed in the center of an extraction column, centrifuged at 12,000 Gs for 1 minute. 1,500 microliters of wash buffer were added. Wash buffer, the stock was supplemented with 64 microliters of ethanol, 96 to 100% previously, and centrifuged at 12,000 Gs for 1 minute, centrifuging rule. It was centrifuged 3 minutes to remove residual wash buffer. The column was changed for a recovery pipe. The liquid was recovered and stored for later use at minus 20 Celsius degrees.
Verification of purity was done using two techniques, by spectrophotometry and an agarose gel at 0.8%. Measurement of concentration of purity in the spectrophotometer were prepared and measured with 1 milliliter of 1 to 100 of purified and unpurified DNA in the spectrophotometer at wavelengths of 260 and 280 nanometers to determine index quality. The electrophoresis in an agarose gel at 0.8% Campbell's 5 mm was prepared. Then load the following samples: 5 microliters of marker 1 kilobase, 5 microliters of purified DNA sample. The conditions are 120 volts for 45 minutes.
The PCR product of ZFXY gene was successfully purified from the gel. In Figure 1, it can be easily identified in its corresponding site according to its size, between the 400 and 500 base pair bands of the 100 base pairs in vitro DNA ladder. In Figure 2, it can be seen the gel from where it was taken the PCR product in well 4. On Table 1, it can be observed the concentration of the purified sample, its DNA and protein absorbance on the quality index. The quality of the purified PCR product is high. The quality index resulted in 25.6609, which reveals the important low content of proteins in the purified sample obtained with the kit. This makes sense when analyzing that most of proteins when present in nucleic acid extractions remain in the upper part of the gel, and that is why it can be said that the sample was repurified. Compared to the original sample, in this purification there were not observed unspecific fragments like those in the initial gel. No degradation, protein contamination, and salt content was obtained in any of both gels, the one of the original sample and the purified one. It is important to analyze the sample purity by spectrophotometry and electrophoresis because with the first one we can identify the protein content index, but impurity is also referred to salt content, unspecific fragments content, and degraded nucleic acids. The yield obtained is high, and this is demonstrated with the elevated quality index acquired, the absence of degradation, and the absence of unspecific DNA fragments, and the high concentration reached.
The critical points when purifying nucleic acids from a gel are: cut the gel with the least possible agarose to make it easier to remove it. Effective fusion of the agarose for adequate removal. Work with sterile material and gloves to avoid contact with nucleic acids nuclease. Use wash buffer and ethanol to eliminate salts and precipitate nucleic acids respectively. When working with silica columns, it's important to use an elution buffer because ions of it interact with charged groups of the nucleic acid or ionic exchanger respectively. First, they elute from the column positively charged molecules that do not bind to the stationary phase, and subsequently adding the elution buffer elute molecules with little negative charge and then the higher net negative charge.
When purifying DNA bands from a gel electrophoresis fragment, it is important to cut the least amount of agarose possible, englobing the DNA fragment of interest, so that less time will be needed regarding the agarose melting process in order to recover nucleic acids for a later elution process. Another important step to remember is that when having a higher salt concentration, it's preferable to apply a silica column purification methodology due to its ability of interacting with DNA, avoiding possible interactions with water and alcohols. The L3 buffer and the washing buffer are needed to get rid of other cellular components that will interfere with molecular biology reactions. In order to confirm nucleic acid's quality after having executed purification in a silica column, it's important to come up with a new gel electrophoresis so that we can confirm that the gel obtained is high, and this was demonstrated with an adequate technique due to a single marked DNA band with the absence of salts and protein contamination.